So I skipped over this earlier, like I said, the nature of antigens. So remember, antigens are, okay, very unhappy computer. I'm sorry, I didn't do it, I promise. It's beeping at me. I don't know where the beeping's coming from. Maybe it's the internet. Well, you know what? Mute usually works. There we go. Because I'm recording. All right. No idea. Okay, so foreign substances, and it can be anything that your body just, as your immune system is recognizing as not you, right? So it even can be something that's you, that your body is mistaking as not being you, right? Could be an antigen. But antigens, as far as it relates to especially antibodies, there's complexity to them, all right? So a bacteria, for instance, is made of several different parts, right? Lots of different molecules on its surface and even within it. So when our body responds, it is actually responding at a molecular level, right? To molecules, even small components within a protein. So that component of an antigen that our immune system is actually interacting with and detecting is referred to as antigenic determinants or epitopes. So these are pieces of the antigen. So remember, the word antigen actually comes from antibody generators, right? So these foreign substances caused our body to produce antibodies. That was our most common immunological response that we could measure. But we now know that we do respond cellularly too, right? We have cytotoxic T cells, we have natural killer cells. Antibody production isn't the only thing we do in response, but it is a very common response to an invader. So some immunologists use the term immunogen instead to be more broad in saying that, you know, it's something foreign that's causing our immune system to respond. It's not very popular to use the term immunogen, right? Still mainly in literature and stuff like that, you're still going to see antigen. So I'll stick to calling it antigen, right? But just informing you guys that, you know, there is talk about trying to change it to saying immunogen. It's one of those things I think would take a really long time for that change to happen. So proteins, polysaccharides, right? We're talking about molecules, complex molecules. Right? Especially ones that are highly repetitive. We can respond to them in a particular way that's called string response. Right? Because they're kind of strung together, very similar repetitive molecules. Where lipids and nucleic acids, not so much, right? Lipids are repetitive, but also not very different from one another, where you can have quite a bit of variation among proteins, because remember, proteins are made out of what? Smaller monomers. There's 20 different naturally occurring components that we put together to form proteins, amino acids, right? So we can get a lot of variability depending on what amino acid out of the 20 and what order they are in, right? You can get a lot of variability with proteins. There's lots of sugars out there too. Right, so you can get lots of different combinations of sugars. You can get a lot of variability. But also they tend to somewhat be repetitive too, sometimes. Lipids and nucleic acids, not really much variability, right? Nucleic acids, we have 5 different nucleotides depending on whether you're talking about DNA or RNA, right? A's, G's, T's, and C's, A, U, G, C. That's it. That's the only difference in those molecules, right? And then, as I said, lipids, right? Lots of carbons and hydrogens and not a lot of variability. So recognizing this antigen is directed at pieces of the antigen. So when we say, you know, you're allergic to penicillin, which is an antibiotic, right? This is a molecule, right? And it's probably some component of the molecule that your immune system is recognizing as foreign. It's not necessarily recognizing the whole molecule, right? It's recognizing probably just a piece of it. For bacteria, too, you're recognizing different pieces of it. So when we look at an antigen, the whole thing, like a protein, so you see this globular protein that's made of lots of amino acids, our immune system will respond by making antibodies is one of the ways that we can respond. Antibodies bind to specific epitopes. So in the case of this particular protein example, you'll see that this section, right, these groupings of maybe 10 amino acids, is being recognized by this specific antibody labeled 1 and colored in red here. That antibody can't bind to any of the other sites on this antigen. It's just recognizing that one small piece, that one epitope or antigenic determinant. And then a whole different B cell made antibody 4, and a completely different B cell made antibody 3, and antibody 2. These are all made by completely different B cells. They're completely different antibodies as far as their antigen binding site, these variable regions of their heavy and light chains, and they bind to only specific points, antigenic determinants or epitopes on this protein. So you'll actually produce a lot of different antibodies sometimes to a particular antigen, not just one. For testing purposes, remember I said we can use antibodies for testing purposes, like when we're trying to figure out what red blood cell type you have. They can purify out a particular antibody, like so just all antibody 1. Those are called monoclonal antibodies, and those are kind of expensive because they're hard to, not necessarily easy to obtain, right? But of course, when we have a response, we don't want to just throw one antibody, right? We want to make as many antibodies as we can that can bind to as many sites on this antigen as possible to be able to tag it for destruction. So remember last time we talked about a lot of the different outcomes that will happen when an antibody binds to an antigen. When you have this immune complex, an antigen and an antibody binding with each other, right? So we had that cross-linking that happens to help clog them up, right? Keep them in a particular area. You had prevention of adherence and immobilization, tagging for destruction by phagocytes, opsonization, activating complement, right? Neutralization of viruses and toxins, antibody-dependent cellular cytotoxicity by natural killer cells. So, these antibodies are pretty amazing molecules in helping us specifically identify stuff that's foreign and needs to be addressed by our immune system. But any number of the ways, right, that antibodies can interact with our immune system. Oops, I'm hitting enter and I want to fast forward. Okay, so looking at a bacterial cell, same thing, right? So this is even more complex, right? But again, it's going to have a lot of different type of molecular complexes on its surface. So our body could potentially produce numerous different antibodies to specific sites, to specific antigenic determinants or epitopes on the surface of a bacteria. Same is true for viruses, anything foreign, right, that gets into our body. Make sense? Right. So how is it that we can make all these different antibodies? Remember last time when we went over the structure of an antibody, we talked about the heavy chains and the light chains, and we talked about those variable regions. Those regions that are different for each antibody that our body produces. So, as I said before, right, proteins, we get a nice strong response, right, because they're highly variable molecules. Lipids, not so much. So we did this last time, and I'm going to really mess you guys up, and actually I'm going to fast forward while we're on the topic. Where is it that I want to talk about? It's way at the bottom, lymphocyte development. So, slide 60. So, in our body, each one of our B cells, and this happens where? Where do the B cells develop? Where do they form their receptors? Bone marrow, right? This also happens for the T cell receptor, right? And I'm having an animation that I'm going to post for you guys. If it isn't already posted, I'll look at our list. That shows T cell development, and where does that happen? Right. They start in the bone marrow, right, and then they travel to the thymus, right? And so their receptor checking and all of that stuff, right? And remember, there's two different types of T cells. All of that happens within the thymus, right? And that's why they're named T cells. So this process, as I said, we're going to look at antibody diversity, but this is true too for T cells. So for antibodies, remember we have two chains, right? One that's called, it's short, that's the light chain, and the other one is the heavy chain. So for in your DNA, within your B cells, you have a whole bunch of segments of genes that code for making variable regions, right? That part of the light chain that's going to interact with the antigen. So there's what? How many different antigens out there in the world? Millions upon billions, right? And is that number stagnant? Do viruses stay the same? Do bacteria stay the same? No, they're always constantly changing, right? So if we had encoded in our DNA, kind of like how we have for our innate immune response, right? In general, bacteria have peptidoglycan, right? So having a receptor against peptidoglycan, that general substance, is beneficial for us to keep around. But could we make, say, an antibody specifically against a specific strain of bacteria? Yes. But if we kept all that information in our DNA, all by itself, that would be millions upon billions, right, of pieces of DNA. That takes up a lot of space and takes a lot of energy to maintain, right, trying to store all that information. So instead of storing it in discrete increments like we do for our receptor for peptidoglycan, instead we do what's called gene rearrangement. We have a whole bunch of regions that are within the DNA that code for different variable regions. So notice 1 through 5, it has choices, right? Which one is it going to pick? Which one is the B cell going to pick? So you can almost say your B cells play the lottery all the time, right? They're picking their segments. And they're going to pick, and notice here, between 100 and 300 different segments they could choose. Then there's a joining segment that they'll choose, and then the constant region. Remember, the constant region is going to allow it to bind to the heavy chain. Now, for heavy chains, they've got an additional variable region. They've got their variable region, they've got their D region, and their joining section. And then they have their constant region, but they have 5 different constant regions, because you guys remember we have 5 different classes of antibodies. And the heavy chain is what is going to give you that stem portion, that FC portion that's going to determine which antibody the B cell is making. So what's the first one we always make? So remember, this is Greek letters. So this is M, right? Mu. Gamma, which is G. Delta, which is D. Epsilon, which is E. Alpha, which is A. Right? Which one do we make first in an immunological response, and is the only one that the fetus can make, and it does not leave the bloodstream? M. Notice how it's first in the list, right? What's the one that can cross the placenta? Is in the monomer form. That's the other thing. What about that M? How is it secreted when a B cell makes it and secretes it? What form is it in? Is it just one little Y? No, it's mega big, right? It's 5 Ys, a pentamer, hooked together. IgG as a monomer, right? And it has the UV link ability of crossing the placenta. Where else is it found? Women produce it at a special time. What breast milk? The first breast milk, which is called the colostrum, right? Only then. IgE. When is that one typically produced? Allergies, right? Remember, Erica hates that one, because Erica has horrible allergies. Lots of IgEs, especially against grass. Alpha. This one is secreted as what? What form? Is it just a single Y? No, it's two of them. It's a dimer, right? And it's secreted. So what secretions do we produce that have IgA in this dimer form? Mucus, tears, saliva, and breast milk, right? Always in breast milk. So when the B cell is developing in your bone marrow, right, it's literally rearranging its DNA, right? So in its DNA, it has this code, and it's picking its lottery numbers. So it has special proteins that are literally going to rearrange and cut out and pick specific portions of these genes within its DNA. Once it does that, it can't revert back. It literally throws away the extra stuff. Okay? So now it's stuck, right? So for this guy, he picked V2J1. That's his number from now on for his whole life. For its heavy chain, and it's going to make the two proteins, right? Messenger RNA make the two proteins. Now for the heavy chain, right, it's going to pick, and in this case, we're going right to IgG, right, gamma. Produces the two. They're going to bind together. That's B cell number 1. B cell number 1, this is its chosen path. It does not change, right? Its DNA ever again. Well, I shouldn't say. It doesn't drastically change it. It can't pick new numbers, right? It's stuck with these numbers, with these sections. It can tweak them a little bit, but he's stuck with these segments of DNA that they've picked out. Now, a different B cell in your bone marrow is going to pick probably different numbers, right? And that B cell is going to therefore be different, right? Its variable region is different. And we even have variability because we've got the combination of heavy chains and light chains, right? So we have variability within the light chains, we have variability within the heavy chains, and then we have that combination of the variability between the two proteins. Make sense? So that's how your body makes all these different antibodies. Your B cells are checked out in your bone marrow to make sure they're not recognizing self. If they do, we eliminate them or we turn them off, hopefully. So that now we have ones that could potentially interact with and recognize a multitude of different antigens out there in this world. Same thing happens for the T cell receptor, right? Their receptors are two proteins. Not as complicated, right? This is still two proteins, but we make two copies, right? So we end up with two identical mirror image arms on an antibody. So in the objective it says gene rearrangement, right? So that's where they're picking these different segments, right, within the DNA. Right? So just like when you hand in your lottery numbers, when you pick those, you can't change them. The combination, right, when we refer to combinational associations, that's the heavy chain and the light chain, creating that different combination. So even if a B cell picked the same numbers for their light chain as another B cell, they probably aren't going to pick the same numbers for their heavy chain. So in that combination, you're going to end up with something different. Imprecise joining is another component where these segments, say V1 and V2, when they join to, say if they put V2 and J1 together, even another B cell that chooses those same ones, the way they put those two segments together is imprecise. So they're going to end up with different overlaps and underlaps of the code. So even if they pick the same number combination, it's still going to be different. Does that make sense? That's the toughest one usually first. Kind of like a finger. I don't know. I can't think of an analogy for this one. I keep thinking if anyone comes up with one, I need a good one for this one. I can't find a good video out there either about it. So that's how we get variability, right? How we produce all these different B cells in our body. Each one of your B cells is a unique, special little guy, right? When they're born in your bone marrow. And that's what gives us our diversity. So now I apologize because I'm going to rewind again. All right. So now I'm going to go right back to right after antibodies to clonal selection. And again, we're going to focus on B cells here, but I'm going to make sure I have an animation for you guys in regards to T cells. So before we even go through this clonal selection and expansion phase, the B cells and T cells have to make their receptors. They have to be checked out. So once that happens, and so notice I said that we're focusing on B cells, but this happens for T cells as well. As I said, we want to be able to recognize amongst all the B cells a limitless number of antigens that may be possible out there. Each cell is only going to respond to a single epitope. Remember, the antibody or the T cell receptor is only going to bind to one small portion of that antigen, not the whole thing. It's random, right? When they pick what segments of the DNA, that is random. So like when you get a quick pick, when you get lottery numbers, right? It's random. At least you like to think it is, right? So the lymphocytes reside in our secondary lymphoid organs. Remember, lymph nodes, spleen, right? Our mucus-associated lymphoid tissues, our tonsils, all these places. And they're just waiting to find their dream antigen, their perfect match, right? And it's got to be that antigen receptor, that B cell receptor, that antibody on the surface of the B cell or the T cell receptor that's going to recognize. But confirmation from another cell is usually needed, right? Remember we talked about helper T cells helping out other cells. So the B cells will then begin to multiply. In this case, when a B cell multiplies, it makes copies of itself. Exact copies for the most part. So therefore, they're referred to as clones. These guys can leave the secondary lymphoid organs. They can migrate to tissues where infection is. They'll keep responding as long as antigen's present, right? So in the case of cytotoxic T cells, we're going to keep producing them. In the case of B cells that turn into plasma cells and secrete antibody, we're going to keep doing that until the threat is gone. Now, most of our army, most of those cytotoxic T cells, once we've defeated the bad guy, they too are going to die through a process called apoptosis. What's the difference about this type of cell death? No inflammation. No inflammation, right? So once these guys have done their job, we don't maintain them. It's expensive to maintain these cells, right? It takes a lot of energy. Why don't we need to keep them, though? Because remember, with the adaptive immune response, if we've activated B cells and T cells, some of them are actually doing the job, others became what? Memory cells, right? Those are going to protect us if it happens again. So we don't need the workers, right? We just need the memory cells. So this is a good animation, but I'm going to skip over it. I'm going to go to this diagram. So again, stem cells in our bone marrow generate the B cells in the bone marrow. Each B cell is going to go through that process that I said. It's going to pick and make its own special antibody. So you'll notice in this picture, this B cell, its antibody it made is different from this B cell, different from this B cell, and different from this B cell. Right? They coded them different colors and some different shapes. An antigen comes along, say it's been brought there by a dendritic cell, or the antigen by itself has been brought to the lymph node through the lymph, through that liquid that's been absorbed from our tissues. And this B cell comes in contact with the antigen. This B cell recognizes it. These other three are not recognizing it. So this B cell recognizes it. It may get assistance from the helper T cell. And if it's activated, right, if, okay, I recognize it, I get the other signals I need, it's going to move forward. It's going to become activated and it's going to proliferate. So we get a whole bunch of these guys that all can produce this same antibody against this antigen. Against one little piece of the antigen even. B cells differentiate into cells known as plasma cells. These are the effector ones that actually do something. They increase their rough endoplasmic reticulum because, remember, antibodies are proteins. And so they're going to secrete millions upon billions. I mean, literally within seconds they can produce millions of antibodies. But the awesome thing about this, right, and they'll keep doing that until the bad guy is gone, we're also going to produce memory cells in this process, right? That those are going to stick around in the lymph nodes in case we see it again. And the response is going to be that much faster. So a T cell or B cell, a lymphocyte, when it's first born, right, it's naive. It has never seen a bad guy. Once it's seen the bad guy, it's been confirmed, it becomes what we call activated, which means it's going to proliferate. It's going to produce copies of itself. And then it's going to produce effector cells, right? These are descendants of activated cells. They'll produce specific cytokines, right, chemical messages to talk to the different cells of the immune system. Plasma cells are what B cells turn into, and they'll actually secrete antibodies, right? Helper T cells can be activated. That will help out B cells in activation. They'll help out macrophages become activated. Cytotoxic T cells are just that. They're going to go out there and kill cells that are infected. Both cases, we produce long-lived descendants, memory cells, right? So if you activate a B cell, you get a memory B cell. You activate a helper T cell, you get a memory helper T cell. You activate a cytotoxic T cell, you get cytotoxic T cell memory cells. And these are the ones that give us that secondary response. These are the ones we hopefully produce when we vaccinate someone, right? So that our response is swift and quick and specific when we see that antigen again. So we're going to go into the specifics of that next time, right? Right down to the receptors. In the meantime, I closed out my canvas. I'll make sure, right, and I'll post an announcement for you guys for that T-cell development animation. I'd like for you guys to watch that before our next class, if you have time. Don't forget, start working on that discussion board, right? Find some cool immunology stuff. Let me end that.